Hello, everyone, and welcome to your blood bank lecture. Today, we are going to discuss chapter 3, and this chapter is pretty straightforward, and we'll be discussing blood bank reagents. They could be a little complicated and a little different. Therefore, it's important that you pay attention to this lecture and that you let me know if you have any questions. So let's start first. First of all, these are the objectives for the lecture, so I won't go through them right now, but when you have a chance, please go back and read these on your own time. These objectives are actually very good for you because you should use them um, when you are studying for the exam. So if you can answer these questions in the objectives, you pretty much can answer all of the questions in the exam. So these are great tools for you to study and to get prepared for the exam. So obviously in blood bank, when we look at the donor or patient's blood in plasma or serum, the original testing that we're going to use, or the primary test that most laboratories use, is tube testing of the source of the antigens and antibodies with either reagent red blood cells or reagent anti-sera. So any type of reagent that is out there is made by a company with known antigens or known antibodies. So reagents can vary a little bit, and this is what this lecture is all about, to be able to explain them. So blood bank reagents, or the reagents that we use in blood bank, really provide us the tools to detect the antigen antibody reaction. So that is the main principle and why we need these reagents in the blood bank. So there are four basic categories, and I just mentioned them earlier. So we have red cells with known antigens in them, or we have anti-sera with known antibodies. We also have the anti-globulin reagents, which is anti-IgG. Sometimes, or in some literature you'll read, they are referred to as the anti-immunoglobulin reagent or the anti-immunoglobulin G and or complement. So these are these are the components that are in the anti-globulin reagents. We also in blood bank use potentiator reagents, or reagents that will enhance certain reactions with the antibodies. So let's explore all of that. But before we can talk about the actual composition of the reagents, we have to talk about how these reagents are being regulated. We can't just buy anything and then use it in the blood bank. Obviously, there's some sort of regulation and quality control and assurance in the blood bank department. So the FDA provides the regulations for licensing this blood bank reagents. So they license commercially available anti-sera and red cell products. The FDA has pretty much established a set of minimum standards that relate to product specificity and potency. Potency is actually Potency in blood bank reagents refers to the strength of the antigen antibody reactions, and specificity in the blood bank reagents refers to the recognition of the antigen and antibodies to make the antigen antibody reactions. A package insert that comes with every reagent should always be included, and the package insert describes in a lot of detail the reagent, the composition of the reagent, the intended use for the reagent, a summary, the principle of the testing. They also provide you with the procedure for proper use of that reagent, and the specific performance characteristics, and also limitations of the reagent. So what is it not going to be able to detect, and also where should you not use, in what instances should you not use the reagents. So there obviously has to be quality control in testing. So quality control of reagents is it has to be performed every day. So we have to do a daily control check on reagent red blood cells and anti-sera. And as you can see here in the slide, quality control, QC, is testing that we do in the lab to determine the accuracy and the precision of the equipment, reagent, and procedures. Some of the components of QC program include a statement of the criteria for acceptable reagent performance. You always have to document the use of the reagent and the fact that you did a quality control check every morning or at every shift change. And if the QC doesn't come in or something went wrong with the reagent, what are the corrective action? What did you do to fix the lack of performance of the reagent? So all of that has to be recorded as well. So records of quality control testing must be maintained. So you have to keep all of the results, interpretations, the date of testing, and the identity of the personnel that actually perform the testing. So usually in the laboratory, what we'll do is we initial. For me, my initials would be CV. So that would always have to come next to the date that I performed the QC, and next to all of the other documentation that is necessary for quality control. So quality control of reagents, also equipment and procedures, which we'll talk about later on, is very, very, very critical in the blood bank lab as well as it is in other labs. So let's talk about some commercial antibody reagents, right? So we said there are red cell reagents and also antibody reagents. So let's discuss polyclonal antibodies, and then we'll talk about monoclonal antibodies in a little bit. So polyclonal antibodies are actually made from several different clones of B cells that secrete antibodies of different specificities. They are able to recognize multiple epitopes on the antigens or the epitopes that are on the red blood cells. And for an example of that, it could be anti-human globulin. So AHG stands for the anti-human globulin reagent. And so polyclonal reagent or a polyclonal antibody comes from several different clones of B cells that secrete antibodies of different specificities. Monoclonal antibodies, on the other hand, are antibodies that are made from a single clone of B cells that secrete antibodies of the same specificity. They use hybridoma technology. They are able to recognize a single epitope. So examples of monoclonal antibodies could be anti-sera that is made with anti-A, anti-little c, and anti-IgG antibodies. So again, monoclonal are antibodies that are made from one single clone of B cells, okay, that secrete antibodies of the same specificity. So that's the difference between monoclonal and polyclonal antibodies. So this table right here is actually very nice because it shows you the exact difference between monoclonal and polyclonal antibodies. So monoclonal antibodies secreted from a single clone of antibody producing B cells, and polyclonal secreted by several different clones of antibody producing B cells. It could detect monoclonal contains one immune globulin class, either IgG or IgM. However, with polyclonal antibodies, there is a mixture of IgM and IgG antibodies. Monoclonal antibodies detect a unique specificity. They have a unique specificity for a particular epitope, as opposed to polyclonal that has a mixture of antibodies that may be directed at different epitopes of the same antigen. So what are some of the antibodies that we use? I apologize for this. It looks like my table went up a little bit, and it covers some of the wording for the first line, but I will post the correct format for this so that you'll have the right handout. So reagents for ABO typing derive from monoclonal antibody sources and could be blended to create reagents that recognize the corresponding A or B antigens. The reagents that contain IgM antibodies, these reagents for ABO typing contain IgM antibodies in a low protein environment. So for ABO typing, we use the anti-A and anti-B reagents, and these reagents are used to identify the ABO blood type, right? So both anti-sera, anti-A and anti-B, are directed towards specific antigens on the patient's red blood cells. So remember, when we do ABO typing, we need the actual red blood cells, right? And we use anti-sera, reagent anti-A, and reagent anti-B to detect the presence of these antigens on the red blood cells. Anti-A will be directed towards the A antigen on the red cells, and anti-B gets directed towards the B antigen on the red cells. So this is how we know what are the antigens that are present on the red blood cells. Oh, and this table right here shows you the ABO red cell typing with forward grouping. Okay, so these reagents have been formulated to give us a very, very strong reaction. If the antigen is there, it should react with the antibody. So it should form an antigen-antibody reaction. So in ABO typing, if everything is going correctly, you should get about a 3 to 4 plus reaction. When we do ABO typing, we are able to immediately see a reaction in the phase that is called immediate spin. So after we add our red cells and reagents in one tube, we can go ahead and spin it down and then read it right away. And that's what the immediate spin phase is. Confirmation testing for the ABO typing is done to check for expected ABO antibodies, and that's when we do the reverse typing. Remember, in ABO typing, when we do reverse grouping, we're really just doing sort of a control for what the forward typing is. This table right here is really nice because it shows you a summary of the ABO and D typing reagents that could be available for the blood bank labs. With D antigen typing, so the reagents for D antigen come from monoclonal antibody sources, and they could be monoclonal antibody blends or monoclonal polyclonal antibody blends. The reagents can contain either IgM or IgG antibodies in a low protein environment. So we've already talked about our the Rh blood group system in chapter 6, and we already know that the D antigen is not the only antigen that is present in the Rh blood group system. In fact, we talked about the D antigen, we talked about the big C, the little c, the big E, and the little e antigen. So those are the ones that are the most common antigens in the Rh blood group system. However, between all of those, the D antigen is the most important, and we always have to type for D antigen because that is the antigen that is going to tell us if the person is positive or if the person is negative. So AB positive, O negative, A positive, B negative. So that comes from the presence or absence of the D antigen on the red blood cells. This antigen is very important because it has increased immunogenicity, and if there is incompatibility, it can cause a hemolytic transfusion reaction. So the AABB standards for blood banks and transfusion services requires that all of the blood samples are typed for the D antigen. Okay, so you guys already know this from the lecture that we've had on blood on the Rh blood group system. So there is commercial anti-D available. It is combined with the patient's, obviously when we do the testing, we take the commercial anti-D and we combine that with the patient and or donor red blood cells. The agglutination indicates the presence of the D antigen on the red cells, and no agglutination indicates the absence of the D antigen. A negative reagent control is always it's always useful to ensure that a false positive result has not occurred. It is very important that we check for all of this, because if we have incompatibility in a transfusion, it can be a big deal. It can be fatal. So this table right here gives us an example of typing for the D antigen with patient or donor red blood cells. So if the D antigen is there, or if it is D positive, you will have a positive reaction with anti-D, and your control should be negative. If the D antigen is negative, you will have no reaction with anti-D, and the control should be negative as well. If you have a positive reaction with the anti-D and a positive control, you cannot interpret these results. The results of the reagent control should be negative always. So this table right here helps us identify high protein reagents and low protein reagents that could be used as anti-D reagents. So remember, they may be monoclonal antibody blends or monoclonal and polyclonal antibody blends. So with the high protein reagents, they contain polyclonal antibodies, approximately 20% of bovine albumin, and these promote false positive agglutination. With low protein reagents, they contain monoclonal antibodies like IgM or monoclonal and polyclonal blends. It has approximately 6% bovine albumin, and they actually have replaced these high protein reagents because of their specificity to detect the D antigen in the patient's or donor's red blood cells. A low protein control reagent is always available to check for the presence of spontaneous agglutination of patients or the donor's red blood cell in testing. This control, again, should always show no agglutination for you to know that the testing that you have done for your patient or for your donor is valid. So again, the control should always show no agglutination. So these low protein reagents control ensure that typing results are correct. Control should show no agglutination, and false positive agglutinations can result, but these will be due to different situations. So these can help identify whether there is a cold autoantibody and a strong reaction for that, or some sort of protein abnormality present. So when is a separate control for D actually used? So a separate control is used if the red blood cells agglutinate with all of the ABO antisera. And a separate control is not needed if there is a negative result using any of the ABO low protein reagents. So if you have any of the ABO reagents when you're doing your ABO typing that come out negative, that can be used as a control for the D antigen, okay, or for the anti-D. However, you do have to include a control if all of your red blood cells agglutinate with all of the ABO antisera as you're doing your ABO typing. So let me know if you have any questions about this, but I want to make sure that you understand when a control is included with your anti-D testing, and what it means when you have a positive and a negative reaction. Okay, so aside from anti-sera antibodies, we also use reagent red blood cells. Reagent red blood cells are used as sources of antigen in antibody screens. We use reagent red cells in the ABO reverse grouping testing, and we also use reagent red cells when we want to do antibody identification tests. Okay, so commercially available red blood cells or reagent red blood cells have known antigens to confirm the presence of antibodies in the patient's serum or plasma. Does that make sense? I think it would. So these manufacturers are able to provide us in the blood bank lab with reagents of red cells that actually have the known antigens so that we can identify the unexpected or missing or whatever type of antibody we're trying to find in the patient's serum. So we use we typically in the blood bank you follow the procedure that the manufacturer includes with the reagent. Some of them will follow the ABO serum testing. So we use serum for reverse grouping with the reagent red cells. We also do some screening tests. So before you can do an antibody identification, you will do an antibody screen just to see if the antibody is present, okay? And then from that, you move on to the antibody identification. And when you do an antibody identification, you're actually identifying. You're not just saying there's a positive or negative antibody. You're saying this is the antibody that is present in the plasma or in the serum of the patient. So as part of the reagent red cells is the A1 and B cells, right? So do you recognize these two reagents? So these two red cell reagents are actually used for the ABO serum testing, okay? So we'll take the reagent red cells, plus we'll use our patient serum, right? Or plasma, and we want to see we want to double check if the reactions from a forward grouping were correct, and also what are the antibodies that are present in their plasma. So these are the reactions that you should expect with the type A, B, AB, and O. So if you take the plasma and you react them with reagent A1 cells, a group A patient will have a negative reaction with A1 cells, but a positive reaction with B cells. And that's because group A patients have naturally occurring anti-B, okay? So the opposite goes for group B patients, which would have a positive reaction with reagent A1 cells and a negative reaction with reagent B cells because they have naturally occurring anti-A in their serum or plasma. A patient who is AB, okay, will have a negative reaction for both reagent A1 cells and reagent B cells because these patients don't have the anti-A or anti-B present in their serum. People that are group O will have a positive reaction with both reagent A1 cells and reagent B cells because they do have the anti-A and anti-B in their serum because they actually lack the A and B antigens on the surface of the red blood cells, okay? So in doing this testing, you already know commercial A1 cells and B cells are combined with the patient's serum or plasma to determine the reverse grouping of the patient. The patient possesses the antibody directed against the antigen of the ABO system that is lacking on their red blood cells, okay? You'll always make the antibody to the antigens that you lack. Okay, so again, from this is a little bit more about A1 cells and B cells that we use in reverse grouping. These cells can come from a single donor or a pool of donors. These have been resuspended in a 2% to 5% concentration. They are negative for the Rh antigens, big D, big C, and big E, and should not be used if red cells are darkened, um, if there is agglutination in the vial, or if they show hemolysis, because you are not going to get the correct answer. There is something wrong with the reagent, and that is something very important to check before you move on with your testing. So dark red blood cells will be a problem. Any type of agglutination in the reagent vial, or if you see hemolysis, obviously you cannot be used. And these reagents also expire because they're red cells, they're viable red cells. So these will expire. So once they're expired, you cannot use them for testing anymore. Okay, another form of red cell reagents are the screening cells. Screening cells, we're going to talk about that a little bit later when we start talking about antibody ID and identification. Screening red cells are actually used in antibody screenings or for antibody detections to test for unexpected antibodies in the patient's serum or plasma. So we have them available in sets of 2 or 3 vials. Each vial may be from a single donor or from 2 donors that are pulled together. So pooled cells can be used for donor testing, but only single donor vials are used when a person is about to receive a transfusion, okay? So when the person is a recipient, okay, only single donor vials are used. Single donor cells tend to give a stronger reaction, okay? So please put a big star next to this, next to this paragraph right here for screening cells. So each lot of screening cells comes with an antigram that shows the antigen profile. So this picture right here is the antigram, and this picture shows you the antigen profile for these red blood cells. The cells must express antigens associated with the most clinically significant antibodies. So the thing here is there are so many different blood group systems that somebody can have antibodies for. However, we cannot test for all of them. So we test for the screening cells that are regulated by the FDA. So this gives you an example of the antigram that has of the screening red cells that have positive or negative reactions for the clinically important antigens. Okay, so remember, these antigens could be of the Rh blood group system, the MNS blood group system, the P blood group system, Lewis, Lutheran, Kell, Duffy, and Kidd. These are clinically important, and this is why it is important to include them with screening of antibodies. Okay, so the screening cells that we buy from the manufacturer will come with these positive and negative reactions. And every time we use a set of screening cells, we have to use the corresponding antigram for those specific cells. I cannot use screening cells for one set and then take the antigram from another set, because it's going to have a different reaction. Those cells are going to have a different reaction, okay? So screening cells are regulated by the FDA for big D antigens, big C, big E, little c, little e, M, N, S, little s, P1 antigens, Lewis A, Lewis B, Kell antigens, big K, little k, Duffy antigens, which is the FYA, FYB, Duffy A, Duffy B, and the Kidd antigens, which are the JKA and the JKB. These are the most clinically important antigen antibody reactions that we'll find. So what we do is we take the screening cells and we react them with the patient's serum, and then we record all of the different reactions at different phases. Okay, so we'll get into this later on, but the phases will be immediate spin, right? The phases will be 37C, anti-human globulin. You'll always have a control in there, and we'll get into that a little bit later. Okay, so aside from screening cells, we also have panel cells. Okay, so panel cells are similar to screening cells, except they are obtained in vials of 10 or more. Sometimes some panels have more than 10 cells. They will be labeled 1 through 10 or 1 through 11. They are used for identifying antibodies in a procedure called antibody panels. Each lot of panel cells will have an antigram that comes with it that shows the antigenic profile for each of the vials. So each one of our vial red cells will have a different reaction with the antigens that correspond to different blood groups that are clinically significant. So we'll get into antibody screen and antibody panels in probably in a few lectures after this. All right, so let's talk about the anti-globulin test and reagents. So the principle of the test is that we take commercially available antibody that has a specificity towards human globulins, and these are used to agglutinate antibody-coated red blood cells. Okay, antibody-coated red blood cells. This is why we need this, because sometimes our red cells are sensitized. And how do we identify what is going on in those reactions? We need the anti-globulin test, the anti-globulin reagents. So anti-globulin test detects IgG molecules and complement protein molecules that have attached or sensitized to red cells but have not resulted in a visible agglutination reaction. The reagents contains antibody towards IgG or anti-IgG and or complement. So it would have anti-CD3 or anti-C3b. Okay, so one type of the anti-globulin test is the DAT test, and I know you guys have heard about this test before. It's the direct anti-globulin test. So the DAT detects antibody or complement molecules that have sensitized the red blood cells because of a clinical event that occur within the body. So anything that occur within the body is in vivo, okay? So this is a reaction that has occurred in vivo. That is going to be very different from what goes on when we use the IAT, and I'll talk about that in the next slide. In the procedure here for DAT, the anti-human globulin reagent is added only after the red blood cells have been washed, okay? Agglutination demonstrates whether IgG or complement was attached to the red blood cells. So you have to wash the patient red cells before the test, and anti-IgG is added to the washed cells. And if you see agglutination of these IgG and washed red blood cells, it shows or it demonstrates that either IgG or complement was attached to the red blood cells. So these are some examples of clinical examples that would cause the positive DAT test in the clinical condition, for example, of a transfusion reaction. A positive DAT could be caused by donor cells that are coated with IgG. The source of the IgG would come from the patient's or the recipient's antibody. in hemolytic disease of the fetus, a newborn HDFN, the fetal red cells are coated with the IgG, and the source of IgG is the maternal antibody that is crossing the placenta. And in autoimmune hemolytic anemia, there is IgG or C3 or complement on the patient red cells, and the source of IgG is the patient's autoantibody. And in drug-related mechanisms, there's Ig drug complex. There's an Ig drug complex attached to the red cells, and the source of IgG is the immune complex that is formed with the drug. So the other anti-human globulin test is the indirect anti-globulin test, which is the IAT. This detects IgG or a complement to the red blood cells, but it does it in vitro. So what is going on in the tube? And it is a two-step procedure, but the IAT actually requires an incubation step for sensitization, and it is an in vitro test. The IAT is commonly used in antibody screens, antibody identification, and testing of donor and recipient compatibility. And these are some applications of the indirect antiglobulin tests in the blood bank lab. We can do them again in antibody screen. If we do it in an antibody screen, we can detect antibodies with specificity to red cell antigens. We can do them in the antibody identification or the testing, which would identify the specificity of the red cell antibodies. We can use them in cross-matching, which determines the compatibility between donor and patient before transfusion, and for antigen typing, which identifies the specific red cell antigen in a patient or in a donor. And these are all common tests that you'll be doing in the blood bank. So the AHG test can possess sources of error that may cause false positive or false negative test results. So recognition and prevention of these sources of error really help with the correct interpretation of the AHG test results. So this table explains some of the common sources of false positive error in the antiglobulin testing, the AHG testing. So you may get a false positive if red cells are agglutinated before the washing step, in addition of anti-human globulin reagent. And some possible explanations for that are the potent cold reactive antibodies that come from the patient or that have patient origin. Sometimes when we use dirty glassware or dirty tubes, that can also cause an issue. And there can be some particles or contaminants that are really giving us that false positive reaction. Improper centrifugation or over centrifugation is a big issue. So we always have to make sure that our times are set correctly, so and our speeds as well, so that we don't over centrifuge our sample. In this case, if this gave us a false positive reaction with the HG test, the red cell button is packed so tightly on centrifugation that non-specific clumping cannot be dispersed from the bottom of the tube. So that can cause an issue as well. So we have to make sure there is no agglutination before the washing step. And before we add the AHD reagent, make sure that the tubes, if you're doing tube testing, they're clean, they're not dirty, and that your times and speeds in the centrifuge are set in the correct speeds and times, so that we don't have any problems with the red cell button in the tubes. And here are some sources of false negative reactions and the possible explanations when it comes to doing the AHG testing. Okay, so these anti-human globulin reagents or anti-globulin reagents, can be polyspecific or they can be monospecific. Polyspecific AHG reagents are used primarily with the direct antiglobulin testing to determine whether IgG or complement molecules have attached to the red cells in vivo. This reagent contains both IgG and anti-C3D antibodies and detects both IgG and C3D molecules on the red blood cells. With monospecific AHG reagents are used in the investigation of a positive DAT to determine the nature of the molecules that are attached to the red blood cells. These monospecific AHG reagents are prepared by separating the specificities of the polyspecific AHG reagents into individual sources of anti IgG and anti C3D or anti C3b. So, it is required by the AABB that we use IgG synthesized cells as a control system when AHG results are negative. So these are used when added to a negative AHG test. Reagents should cause agglutination. Commercial reagents are typically or mostly type of red blood cells that are prepared with IgG antibodies that are attached to them, and some false negative results um, can also be apparent, and these can be caused by failure to add the IgG reagent or failure of the IgG reagent to react with the sample or failure to wash the red blood cells adequately. So the washing step is very, very, very important when we do our antiglobulin testing. Okay, other types of reagents that we use in the blood bank lab are potentiators. And potentiators are antibodies or their enhancing media that are commercially available reagents that help us enhance the detection of IgG antibodies by increasing their reactivity. So, some examples of potentiators or examples of enhanced media include LISS or LIS, PEG, VSA, and different types of enzymes. LIS is low ionic strength saline, and these reagents help increase the rate of antibody uptake. Polyethylene glycol, which is PEG, concentrates the antibody in the test environment in LIS. Proteolytic enzymes like papain or physin, they actually remove negative charges from the red cell membranes, which reduces the Zeta potential. So it denatures some of the red cell antigens. So the agglutination reactions are going to be very different after you treat your cells with proteolytic enzymes like papain or physin. BSA is bovine serum albumin, and this reduces the repulsion between cells, but it does not shorten the incubation time. Okay, bovine serum albumin. It is available in 22% albumin or 30% concentration. It is commercially available in those percent concentration, and albumin will allow the antibody sensitized cells to become closer together than is possible with just using saline. So it favors direct agglutination with Rh antibodies, and it also enhances the sensitivity of the indirect antiglobulin test. Lectins are also very, very special. Remember, lectins are plant or they come from other extracts. And these lectins bind to carbohydrate proteins of a certain red cell antigen and agglutinate the red blood cells. Although no antibodies exist in these lectins, or in these reagents, lectins can be useful in identifying antigens present on patient or donor red blood cells. So it's not the antibody that is present, or it's not that it's a specific antibody that is inside this reagent. It's actually a plant derivative, or they're like plant extracts, and these extracts, they bind to the carbohydrate portions of the red blood cells and agglutinate the red blood cells. So they're very helpful in identifying antigens that are present on patient or donor red blood cells. So they can be seed extracts or plant extracts that have specificity towards certain red blood cell antigens. And these are some examples of the lectins that are commonly used, and the antigen specificity that they have. So the Dolichos biflorus has a specificity against the A1 phenotype of type A. The Ulix europus has the affinity for the H antigen. The Vicia graminea has specificity for the N antigen, and the Iberia samara has specificity for the M antigen. Okay, so in blood bank, we are lucky enough to have other commonly used tests, or other tests that are used very commonly, other than just doing the tube. And gel technology is pretty much everywhere in every blood bank lab, even though the most common testing procedure will be tube, but that's because it's very inexpensive to use, tubes. They cost a lot less than buying these gel cartridges, and getting the gel centrifuges and things like that. So tube testing will always be sort of like the foundation of the blood bank lab. So gel technology, the FDA actually licensed gel technology about in the 1990s. It was actually developed back in the 1980s, with this technology that uses dextrin acrylamide gel particles, and they actually trap agglutinated red blood cells. So with tube shaking, right? When we do a tube method, we have to do tube shaking to resuspend the red cell button. And these tube shaking techniques to resuspend the red cell button in the tube testing can vary among technologies. So it could be an issue when you do tube testing, because I could shake a tube a little bit harder than the technologist that's working in the second shift or somebody else. The reading can also be subjective, because what I see might not be seen by the person that's going to do the test after me. So JEC technology is actually good because it provides like a stable and defined endpoint of hemagglutination, and it provides a method where objective and consistent interpretations of agglutinations are made. So if you get when you get a 3+ or when you get a 4+, and that's really not going to change between one technologist and the other. However, with tube shaking and tube testing, there's a little bit more subjectivity involved, so that could always be it could always be better to have a method that is more stable across the whole broad the broad spectrum. So with gel testing, gel particles are combined with reagents and are pens in microtubes of plastic cards. Red blood cells or plasma or serum are added to the microtube and allowed to incubate before centrifugation. The large agglutinates are trapped at the top of the microtubes, and that actually shows or signifies that there is a 4+ reaction. Non-agglutinated cells travel to the bottom of the microtube, and that would indicate a 0 reaction. So no agglutination, no reaction at all. So this picture right here shows you the range of reactions in gel technology, which is really nice, and I think it's a lot more comprehensive sometimes than what the tube testing is like. So you can see what the different reactions are between 0 and 4 plus, and you also have a mixed field reaction in which there's a line on the top and a line on the bottom. So other methods of agglutination reactions are the microplate testing methods. And these are 96 well microtiter plates that actually replace the tube testing. It pretty much applies the same principle as the tube test method, but a concentrated red cell button indicates a reaction. In a dispersed cell, right? Dispersed cells indicate no reaction. So you can see that here in the picture are the reactions of the microplate testing. You see a negative reaction, and then you see a concentrated button, which is a positive reaction. A concentrated button of red blood cells, and with negative, there is a smooth suspension of red cells or a streaming pattern of red blood cells when the plate is when the plate is placed on an angle. And microplate testing were discovered in the 1960s. So some labs have it, some labs use gel, some use tube as well. The next method that we use in blood bank is the solid phase method. And this is another method that's used in blood bank for red cell adherence. So they actually sell solid phase, commercial solid phase test procedures. And this is for the detection of both red cells and platelet antibodies since the 1980s. This microplate this technology of solid phase actually uses the microplate test wells with immobilized reagent red blood cells. Solid phase technology could be used for antibody screening, antibody identification, and also for compatibility testing. So one of the major advantages of solid phase technology is that there can be standardization. There is, one result is pretty much the same for another result, and it is a stable, defined endpoint technology. So what I see as a positive would be what you see as a positive if you were doing this test, after me or before me or whatnot. It is also suitable for automation. And blood bank instrumentation for solid phase is available it's an available technology for blood bankers. So in this solid phase technology, solid phase red cell adherence method, the antigen or the antibody gets immobilized to the bottom and in the sides of the microplate wells. In a direct test, the antibody is fixed to the wells. Antigen positive red cells from donors or patients, they adhere to the sides and to the bottom of the wells. Antigen negative red cells from the donor or patient sources, they settle to the bottom of the well and form a red cell button after centrifugation. So that is for direct testing. And for indirect testing, the red cell membranes are bound to the wells. An unknown patient or donor serum is added in a lab to react. So this step allows for the capture of the IgG antibodies from the patient or the donor serum to the red cell membranes. There's a washing step with that. So the washing step actually removes all of the unbound IgG antibodies, and then you add anti-IgG coated indicator cells. In a positive indirect test, the indicator cells adhere to the sides and the bottom of the well. In a negative indirect test, the indicator cells settle to the bottom of the well and form a red cell button after centrifugation. So this is a little bit different than the microplate testing method, and obviously from the gel and tube method. And so the positive and negative reactions that are interpreted a little bit different. So cells that adhere to the sides and the bottom of the wells are positive, and cells that settle at the bottom of the well of the well are negative. And you can do this in a direct manner, or you can do it in an indirect manner. And more information about your different types of testing is found in section 6 of chapter 3 in your blood bank book. And it actually gives you more pictures and a nice explanation of the different testing that's available in the blood bank. So with that, I will end this lecture. And if you have any questions, please let me know. And one of the assignments is going to be for you to do the critical thinking questions, and we'll go over those together before the exam. Please let me know if you have any questions. As always, I am here to answer to the best of my ability. Thank you.